Hi, my name is Caitlin Schuhart, and what I researched was the effect of HPV early protein E1 suppression on the production levels of HPV oncogenic E6 and E7 proteins. So, the importance of finding treatment for cervical cancer, there are a lot of them. Cervical cancer is one of the top women's cancers worldwide, and every year there's about 570,000 new cases, with 274,000 of those resulting in death. In the United States, there's about 13,170 invasive cervical cancer diagnoses every year, with 4,250 of those resulting in death. It's really hard to catch cervical cancer without routine testing because there are no symptoms typically until it's advanced or invasive. Another issue with it is in many countries there is not routine testing available for cervical cancer. And since there's no routine testing, most of those people will not go get checked out until they have symptoms, which again means it's probably advanced or invasive. The human papillomavirus, HPV, it's found in nearly all cervical cancers, a little over 99%, which makes it really relevant in research. So, what is human papillomavirus? Well, it is a small, non enveloped icosahedral double stranded DNA virus that will specifically infect humans. There are versions of this that do infect other animals, but this specific one, HPV, is only humans. And as I said, it's detected in 99% of all cervical cancers. There are about 160 different variants of HPV, and they are split into two different groups, low risk and high risk. Low risk HPV strains typically do not cause any symptoms, or they can cause warts, and typically the low risk HPV strains will go away on their own. And a lot of the time, it's without the individual even knowing they had it in the first place. The high risk strains also typically go away on their own without an individual knowing, but when there are a lot of cases where it will linger around, or it will start to cause changes in the cervical tissue, which is known as dysplasia. And it was a really interesting fact that about 10.2% of women who have a normal cervix on exam actually do have HPV. So, the human papillomavirus DNA genome is divided into three different domains. So, the first domain is the non coding upstream regulatory region. The second domain is a region with 6 genes that encode the early proteins, which are E1, E2, E4, E5, E6, and E7. And with the diagram on the right, you can see what each specific one does. And then the third domain is a region that contains 2 encoding genes. It's L1 and L2. L1 encodes the major protein of the viral capsid, and L2 is the minor protein. So for human papillomavirus to get into the cell and start wreaking havoc in the cervix, first it must be able to infect a cervical cell in order to replicate, and if it can't replicate, it won't survive. So it has to enter a cell that can replicate, and in the cervix, that is only the basal cells. And then, so for HPV to enter the basal cell, it has to attach to surface cell receptors, specifically L1 and L2, which is what initiates the infection process. Once HPV has entered the cell, that L1 and L2 protein will disassemble, and then the HPV DNA will move into the nucleus of the cell. Once it is in the nucleus, E1 and E2 proteins from the second domain I talked about earlier will become active. So E1 is important because it activates a viral DNA replication, and E2 is really important because it initiates transcription. And specifically, E2 will produce E6 and E7 proteins. E6 and E7, they'll inhibit tumor suppressor genes, and these are the main oncogenic factors in cervical cancer. They cause the most damage. Specifically, E6 will disrupt the P53 tumor suppressor pathway, and E7 disrupts the retinoblastoma or RB pathway. So therapeutic targets. Vaccines actually target L1 and L2 protein, which stops the HPV from getting into the cell in the first place. And then another target that a lot of research looks at is the early proteins. I focused on E1, and E1, there is a drug that inhibits its ATPase, which is biphenylsulfonic acid. There's also a drug, repaglinide, that inhibits the E1 and E2 interaction. If E1 and E2 cannot interact, there will not be any DNA transcription, which means no more E6 and E7 are being produced. So my rationale was if E1 is blocked by biphenylsulfonic acid inhibitor, it would make sense that there would be less production of the E6 and E7 over time. If E1 and E2 interaction is blocked by repaglinide, then it would make sense that there would also be less production of E6 and E7 over time. Given this, it led me to my hypothesis that the suppression of the E1 early protein of the HPV genome by the combined treatment of biphenylsulfonic acid inhibitors and repaglinide will decrease the level of the early proteins E6 and E7 production more than either of those treatments alone would. So my experimental design, I'll test that proposed hypothesis in 3 different ways. Treatment with the E1 ATPase inhibitor, treatment with repaglinide, the E1 E2 inhibitor, and then treatment with both of those. And then in this experiment, HeLa cells will be used in all of them. So the samples of the HeLa cells kept in culture plates using agarose gel will be infected with HPV6 and upregulated with YAP1, which is a protein that allows for faster cancer progression. HPV6 is specifically chosen because it's known for its susceptibility to those 2 drugs. And then the samples will be in a lab at 25 degrees Celsius temperature controlled room with a fixed 12-hour light cycle. Treatments via direct injection will begin 8 days after infection in order to allow for the cervical tumor to establish. Using flow cytometry, the amount of E6 and E7 will be measured and documented. So the first experiments will be the control with no treatment, and then there'll be 2 experiments with the ATPase inhibitor and then the repaglinide, and then the final one will be both. So as you can see here, the control with nothing being treated, there will be a much higher E6 and E7 production since nothing's there to stop it. And then with the ATPase inhibitor and repaglinide, that they will suppress it a little bit. There will still be some E6 and E7 production, as it will not stop all of it, but it will be greatly decreased. And then treatment with both the ATPase inhibitor and repaglinide will really drastically decrease the amount of E6 and E7 production over time. So the conclusion is the results of the study do support the hypothesis that E6 and E7 production levels decrease when the E1 activity in E1 and E2 interaction is inhibited. Application of this treatment would be very beneficial for many cervical cancer patients and could potentially save many lives. This is because if E6 and E7 is their production is decreased over time, there will be less cancer growth and it will be easier to treat. Thank you.